Good day everyone, my name is Johanna Binti Johari and today my group members and I will be discussing an innovative study on the forensic analysis of tears using ATR-FTIR spectroscopy. While blood, semen and saliva are commonly analyzed body fluids in forensic investigations, tears have been largely overlooked despite their potential evidential value. This research employed three main key aspects such as the detection and identification of tear stains using ATR-FTIR spectroscopy, the discrimination of tears from other common body fluids found at crime scenes, and the potential use of tears of age estimation of evidence. The study employed advanced spectroscopic and chemometric techniques to analyze tear samples collected from 24 healthy individuals. The researchers also compared tear spectra with those of blood, semen, saliva, urine, and water. This work is significant because it introduces a non-destructive, rapid and accurate method for identifying and analyzing tear evidence which could prove valuable in certain criminal investigations. It also demonstrates the potential of ATR-FTIR spectroscopy combined with chemometric analysis as a powerful tool in forensic science.
Moving on to the method. The first step for the method is sample collection and preparation. Tear samples were collected from 24 healthy individuals aged between 20 to 40 years old, all of whom had no known ocular disorders. The tears were deposited on two types of substrate, white cotton fabric and tissue paper, to simulate common real-life scenarios. To induce tear production, methanol fumes were employed. Samples were collected directly onto the substrates, ensuring there was no contamination by using gloves during the process. Each collection yielded between 100 to 500 microliter of tear fluid. The infrared spectra of the tear extracts were acquired using a Perkin-Elmer Spectrum 2 FTIR spectrometer equipped with a 2 mm diamond crystal ATR accessory. The spectral data were collected over a range of 500 cm -1 to 4000 cm -1 with a resolution of 4 cm -1 and 64 scans performed for each spectrum to ensure accuracy. Before each sample application, the crystal was meticulously cleaned with 70% methanol to avoid any cross-contamination. The third step is spectral pre-processing. Several pre-processing steps were undertaken to prepare the spectral data for analysis. These included baseline correction to remove any background noise, range normalization to standardize the spectra, ATR correction to compensate for the accessory's influence, and smoothing using the Savitsky-Golay algorithm to reduce spectral noise. These pre-processing steps were crucial in minimizing noise from external light sources, manual vibrations, and potential human error, thereby enhancing the quality and reliability of the spectral data. The last one is chemometric analysis. For the analysis and discrimination of the tear samples, principal component analysis (PCA) and linear discriminant analysis (LDA) were employed. PCA was used to detect and distinguish tear samples from other body fluids and water by reducing the dimensionality of the spectral data and highlighting the most significant variables. LDA further classified the tear stains based on the spectral characteristic at different time points, enabling the determination of the age of the stains. This chemometric approach provides a robust framework for the detection, discrimination, and aging of human tear stains using ATR-FTIR spectroscopy.
Next, let's discuss the results obtained by this study. All in all, this study uses three different methods of analysis. The first one is ATR-FTIR spectroscopy. The second one is principal component analysis, PCA. And the last one is linear discriminant analysis, also known as LDA. All this analysis was done to distinguish tear stains from other bodily fluids found at crime scenes, as well as to classify the samples of tear stains from different time points. Firstly, in order to identify a stain as a tear stain, we must first know the characteristic peaks in the spectra of tear stains that would discriminate them from other bodily fluids. Based on this standard spectra of tears, there are 5 distinct peaks that could be observed in determining tear stains. The first one is the methyl and methylene stretching located at 2922 cm1, followed by CH2 stretching of lipids at 2852 cm1. Next distinct peaks of tears also include the 1737 cm1, which belongs to the fatty ester linkage C double bond O, a 1546 cm1 peak formed due to the presence of amide 2, and lastly a phosphate ester bending and stretching at 1247 cm1. These peaks were all previously reported as the characteristic peaks of tears in a study done by Nagase et al. And Elkins. Moving on, this figure above shows the ATR-FTIR spectra of tears in comparison with other bodily fluids. The study found that among all the characteristic peaks for tears reported by Nagase et al., only one peak was not identified in any of the other bodily fluids. While every other peaks mentioned previously were found present in the blood, saliva, and semen, the sole peak that was not identified in any other bodily fluids is the C double bond O fatty ester linkage peak located at 1737 cm1. After ATR-FTIR, the study then analyzed each bodily fluids using the principal component analysis (PCA) method in order to explore any significant difference between them. It was observed that while blood, semen, and urine showed a significant difference when compared to tear stains, saliva was very close in proximity with the tear stains. Hence, further classification were required for the discrimination of tear stains from other bodily fluids. By implementing the linear discriminant analysis (LDA), the study successfully differentiated tear samples from all of the other bodily fluids, including water, with 100% accuracy. This proves that the LDA-based 3-dimensional classification plot is conclusive and is more suitable for the classification of bodily fluids. Hence, this brings us to the last part of the study, which is tear stain age estimation through LDA classification of tear stains from different time points. Result exhibited had 100% accuracy, and as observed, the tear stain from different time points were very far apart from each other. This indicates that tear stains from different ages can be easily identified through LDA classification.
As for conclusion, the identification and discrimination of rarely found body fluids at the crime scene are as important as for the other common ones such as blood, semen, and saliva. In the present study, tear stains were identified and discriminated from the IR spectra of other commonly found body fluids, blood, semen, saliva, and urine. Unlike blood, semen, or urine, tears have near transparent visibility, which makes the location of these stains significantly difficult. To overcome this problem, an alternative light source was used, which successfully identified the stains on porous substrates, which was further confirmed through the IR spectra. The study further investigated the potential of the trials as the age estimation marker at the crime scene. The obtained spectra from all the body fluids were analyzed by PCA-LDA, discriminated them in their respective classes with 100% accuracy. These results are highly significant for the discrimination of tears at the scene of crime by using ATR-FTIR spectroscopy from the other fluids. Further, in this study, the spectral changes in the tear stain over a longer time period were observed to investigate the potential of this fluid as an agent marker at the crime scene. That's all from us. Thank you.